My name is Elika Shivasmore from the Advanced Centre for Biochemical Engineering at UCL, UK. This work addresses bioprocessing of tobacco plants, and what is particularly interesting about it is that tobacco is usually associated with negative connotations of smoking. However, the plant itself nowadays can be used effectively in production of products such as therapeutics and vaccines. The challenge which remains to be addressed is the way you can manufacture the product of interest in this particular plant in a way where it's going to be as clean as possible so that you can meet the specifications required by FDA. The first step that we need to consider is how to actually break material down and extract the product of interest. Now normally if we do that in the leaves carry a lot of phenolics which are a major contaminant and needs to be removed. However, in this study, which is a continuation of some of the previous work we carried out and published, we are looking at an alternative location for the product, and that is the roots of the plant. And I now hand over to my colleague Dr. Salih Hassan, who's going to be telling you something about how you can use very small quantities of material, which at this stage would be small because it's exploratory, and extract the material in an efficient way so that we can then scale this up and put it into a manufacturing facility.
Just to follow on from Ellie, here's an example of transgenic tobacco plants used in these experiments.
The roots of these plants were about 4 to 6 centimetres in length and were generally white or yellow brownish in colour, although no distinction in root colour were made in these experiments, since it is unlikely during large-scale processing that roots will be separated out into colour. In a previous paper we examined the mechanical properties that characterise these roots via micromanipulation, and this work was also published in Biotechnology Bioengineering. This new work is a follow-on paper examining the mechanical breakage of transgenic tobacco roots in an ultra-scale-down shear device. The device shown here was designed specifically for this purpose with scale-up in mind, potentially also to mimic a large-scale homogenizer. A large impeller-to-tank ratio allowed for high radial velocities. The impeller within the perspex chamber consisted of 8 blades, each with a double serrated edge, and was very efficient at root breakage with a fast fragmentation rate. The device was operated at a constant speed for 4 different time periods. Following shearing and centrifugation, the number of intact roots post-shearing were estimated as the mass of roots post-shearing over the initial root mass. Image analysis was used to estimate the remaining root debris, and fragmentation was approximately first order in root number. Since plant-to-plant IgG variability exists, as demonstrated in the leaves in a prior publication, roots from more than one plant were used per experiment, and IgG was expressed as a function of that released by the laboratory gold standard of grinding in liquid nitrogen. By assuming that the main mechanism of root breakage was via inertial forces, in particular by collisions with the impeller, we established an equation that can predict the root breakage and IgG release in this ultra-scale down shear device, and this can potentially be used at large scale. 36% of the roots remained unbroken under prevailing conditions. In spite of the fact that 36% of the roots remained unbroken, we were able to release equivalent quantities to methods that have been established to the laboratory standard. It was assumed that IgG release was proportional to the number of fragmented roots, and the proportionality constant was found to be 2. Increase in IgG level continued slightly after maximum root breakage, suggesting a process of leakage from the broken root segments.
We would like to acknowledge UCL, Birmingham and St George's and the EPSRC for funding the project. Thank you for listening, and to find out more please read our publication in Biotechnology Bioengineering.